Alright guys, welcome back to the Road to Success MCAT test prep series. This video, I will be talking about development. Development serves as essentially a part 2 or an epilogue to what I discussed in the last video, which was a reproductive system. This video, I'll be talking about fertilization, which is essentially where we pick up from last time, and it'll be moving forward with differentiation and specialization of cells. This will involve changing the cells, or it'll involve creating stem cells. I'll be wrapping this up by talking about cellular regeneration and cellular senescence, or natural aging. This isn't as important as the reproductive system video, but it is important as it fills out all the nooks and crannies that you might have had to experience from last week's video. So that being said, let's jump right into it, and I'll start off by talking about fertilization.
All right, let's start off with fertilization. Fertilization occurs in the widest part of the fallopian tube, called the ampulla. When the sperm meets the secondary oocyte of the fallopian tube, it binds to the oocyte and releases acrosomal enzymes that enable the head of the sperm to penetrate the corona radiata and zona pellucida. The first sperm to come into contact with the secondary oocyte's cell membrane forms a tube-like structure known as the acrosomal apparatus, which extends to and penetrates the membrane. This pronucleus will then freely enter the oocyte, causing meiosis II to complete. After the penetration of the sperm through the cell membrane, the cortical reaction, which is a release of calcium ions, will occur. These calcium ions depolarize the membrane of the ovum, which has two purposes. Depolarization prevents fertilization of the ovum by multiple sperm cells, and increased calcium concentration increases the metabolic rate of the newly formed zygote. Now, I want to have a quick discussion about twins. Twinning occurs through two different mechanisms. Dizygotic twins, or fraternal twins, form from fertilization of two different eggs that are released during one ovulatory cycle by two different sperm. Each zygote will implant into the uterine wall, and each will develop its own placenta, chorion, and amnion. If the zygotes implant close together, the placentas may grow onto each other. Fraternal twins, I want to point out, since they're not identical, they are no more genetically similar than any other pair of siblings. Monozygotic twins, which are the identical twins, form when a single zygote splits into two. Because their genetic material is identical, their genomes will also be identical as well. If the division is incomplete, conjoined twins may occur. That being said, there are more gestational structures that are shared, and there are more risks for the fetuses as they grow and develop. Now let's move on to blastulation and gastrulation. Several divisions after the initial implantation of the sperm, the embryo will become a solid mass of cells known as a morula. Once the morula is formed, it starts to go through blastulation, where it forms the blastula, which is a hollow ball of cells with a fluid-filled inner cavity known as a blastocoel. The mammalian blastula is known as a blastocyst, and it consists of two noteworthy cell groups, which are the trophoblast and the inner cell mass. Trophoblast cells surround the blastocoel and give rise to the chorion and later the placenta, whereas the inner cell mass protrudes into the blastocoel and gives rise to the organism itself. I know I was just thinking, but I don't have a good drawing of it right here, so I'll give you one right now. It's essentially like this, and essentially just looks like this. So this area right here is the trophoblast. And this one right here is the inner cell mass. But yeah, like I said, the trophoblast will surround the blastocoel and give rise to the chorion and the placenta, while the inner cell mass gives rise to the organism itself. Moving on to gastrulation. Gastrulation occurs when gastrulation occurs with the generation of 3 distinct cellular layers. I believe I have it on the next slide, actually. But yeah, moving on, gastrulation, it forms 3 different layers known as the ectoderm, the mesoderm, and the endoderm. The ectoderm gives rise to all of these structures right here. I want to point out that the way I think of it is I call it the attractoderm. Because it forms parts of our bodies that attracts other people, such as skin, hair, our nose, our mouth, eyes. It forms the brain as well. I don't have that written down here, but it does form the brain. That being said, moving forward to the mesoderm, which is the middle layer. This is the outermost layer, obviously given by the ecto. And then the mesoderm is the middle, endo will be the inner layer. But the mesoderm, it forms basically everything that you need to be able to move your body around. So I call this the mesoderm. So it creates the musculoskeletal system, the circulatory system, the excretory system, gonads, all of that. And lastly, we have the endoderm. The endoderm, it forms the lining of your internal organs, or internal organs. I refer to it as endernal, so you can remember that it's the endernal or the endoderm. That being said, it forms the digestive tract, the respiratory tract, lungs, pancreas, thyroid, bladder, urinal tracts, and liver. Moving on, let's talk about the process of neurulation or differentiation. It forms from the notochord, and then eventually the neural tube, and then the neural crest cells. This process right here is written out for you guys. Feel free to study that, but I do want to point out that the notochord gives rise to the spine, the neural tube gives rise to the central nervous system, or CNS, and the neural crest cells give rise to the peripheral nervous system, or the PNS. Now let's move on to cell specialization. Starting with specification, that's when the cell is reversively designated as a specific cell type. This is then followed by determination, where it starts to commit to that cell type. After determination, the cell is irreversibly committed to a specific lineage. The presence of various or specific mRNA and protein molecules will result in determination. Determination may also occur due to secretion of specific molecules from nearby cells. These cells are called morphogens, and they cause neighboring cells to follow a particular developmental path. Now let's move on to stem cells. Stem cells are very interesting as they are able to essentially become any sort of cell in the body. The rate at which they can become a different type of cell is called potency, and the cells with the most potency are called totipotent cells. Or actually, I'll write it a bit differently. Totipotent cells are able to transform into just about any cell in our body, whether it's an adult cell or a placental cell. So right here, that turns into all cells. Pluripotent is a bit different in that these cells can differentiate into any cell type, except for the ones that are in the placenta. And then lastly, we have the multipotent stem cells, which are able to differentiate into multiple types of cells within a particular group. So they have to commit to a certain group. Now let's move on to cellular communication. There are several different types of ways our cell can communicate with itself, or to each other. Starting off with autocrine cells, this is when the cells use signals to target itself and reinforce as a sort of action or reaction. Paracrine signals act on cells in the local area. Juxtacrine signals do not usually involve diffusion, but they involve a cell stimulating receptors of an adjacent cell. So they're usually right next to each other. Lastly, endocrine cells are the most important in that they are the ones that will target cells that are far away from each other. This is going to be the basis of an entire video I have about the endocrine system. So please take note of the endocrine and look out for it. Moving on to the concept of inducers. Inducers are growth factors, and these are peptides that promote differentiation and mitosis in certain tissues. These often use gradients, morphogens, or molecules that cause the termination of cells diffuse through the organism. Locations closer to the origin of the morphogen will be exposed to higher concentrations, while areas further away will have less exposure. Finally, let's talk about apoptosis. Apoptosis is programmed cell death, and it occurs at various times in development. During the process of apoptosis, the cell will go changes in morphology and will divide into several self-contained protrusions called apoptotic blebs, which can then be broken apart into apoptotic bodies and digested by other cells. This allows for recycling of materials. Because the blebs are contained by a membrane, this also prevents the release of harmful substances into the cell, or the extracellular environment. This is different from necrosis, in which a process of cell death will result or occurs from injury, and those internal substances can be leaked, causing irritation or an immune response. Lastly, we have regeneration and senescence. Regeneration is essentially our body's way of replacing or regrowing certain parts. Some species, such as salamanders and newts, have an enhanced capability to regenerate whatever body part they can, because they have a lot of stem cells in their bodies. When regeneration is required, these stem cells can migrate to the appropriate body part to initiate regrowth. These species are known to go through complete regeneration. This is where lost tissues or damaged tissues can be replaced with identical tissues. That being said, humans and most mammals go through incomplete regeneration, where a new formed tissue is not identical in structure or function to the tissue that has been injured or lost. That's very similar to scarring right here, as you can see right here. This scar, it's probably going to stay for a very long time, and it's not going to blend in with the rest of the skin. Human regenerative capacity varies by tissue type, though. Liver tissue has very high regenerative capacity and can often completely regenerate itself following injury or loss. That being said, the heart, as well as our skeletal muscles, have little, if any, regenerative capacity, and extensive damage to these often causes severe damage to our body. The kidneys have moderate regenerative capacity and are at least able to repair their nephrons after injury to the tubules. Lastly, let's talk about senescence. Senescence is the process of biological aging, and it can occur at the cellular level and the organismal level as these changes accumulate. At the cellular level, senescence results in the failure of the cells to divide, normally after 50 divisions. Research shown that this is due to shortened telomeres, or the ends of the chromosomes. Telomeres reduce the loss of genetic information from the ends of chromosomes, and they basically serve as a cap that prevents DNA from unraveling. That being said, they're very difficult to replicate, and they end up shortening during each round of DNA synthesis. Eventually, these telomeres become so short that the cell can no longer replicate. Some cells, such as germ cells, fetal cells, and tumor cells, express an enzyme known as telomerase, which is essentially a reverse transcriptase that is able to synthesize the ends of chromosomes and prevent senescence. Telomerase allows cells to divide indefinitely and may play a role in the survival of cancer cells. So that being said, that's all I have for you guys today. We covered a lot of interesting topics such as regeneration and senescence, as well as cellular communication, specialization, differentiation, blastulation, and fertilization. This basically serves as an epilogue from the reproductive system video that you might have just seen. I know it was a lot of difficult concepts, but that being said, it's not super high yield, but you still need to know it for the exam, so I'm glad you guys stuck through till the end. I hope you guys learned something from this video, and I hope you guys have a great rest of your day, and happy studying.